Moving into the last topic we have on tissues before we move into organs, because they kind of connect to each other, we're going to finish off with how our tissue regenerates. So that could just be through everyday um, situations, or it could be through some kind of trauma, as in injury or an operation or something like that. So we'll have a look at the process that we go through, which will then into tissue engineering. Looking at something like tissue repair, and remember this can come from everyday, walking around, tear and repair of our different tissues in our body. It's tissue that's already formed, cells that have already formed, that kind of thing. So it's actually repairing what is already there beforehand. So it's trying to restore the structure, I guess you could say, or the architecture of the original tissue, the original cells, so that the tissue can then continue on performing its function. So that's why it's really, really important. You can see we'll go through each of these stages, but having a quick look at the picture in the bottom there, this is an example of sort of a skin cell. We've got the epidermis, so it's the top of our skin there. You can see, hopefully, I'm looking at letter, A. So we're looking at epidermis at the top, that's our skin. So we've gone past, we've had some kind of injury, scratch, whatever, to the surface of our skin. Then if we look down further through the same image, we can see through the different blood vessels, we've got macrophages, we've got neutrophils that are rushing to that site to start our repair. So that's part of our immune system. At the same time, we've got the blood starts to clot within the wound, and that helps to keep out, different pathogens that could get in to infect the body, and also from, too much blood leaking out. So now looking at B on the right-hand side of your slide there, the skin, you can see the difference in the structure. So we're starting to repair that architecture now for that skin tissue or that epithelial tissue. So we've got a scab now forming at the top of the surface of the skin. So again, that then becomes hardened. As anyone that's had a scab would know, it's really important that you don't go picking off your scabs, as much as it may seem like a good idea, but it's there for a reason. It's to allow the skin or the cells underneath the epithelial tissue to regenerate without becoming damaged again in the process of doing that. So after we've had that scab formed, we then get these fibroblasts that come along to the area, and they start to help repair that tissue underneath the surface first before it moves to the top. This is happening. There's two main processes. So we end up with, by the end of our tissue renewal or regeneration, we end up with either our tissue or our cells being regenerated. So therefore the architectural structure of that tissue has been repaired, hopefully back to normal, or it could be that we need to replace a particular amount of epithelial, for example, tissue. So sticking with that epithelial, as you can see in the image, so we have active cells. We'll look at each of these ones later, but when we look at tissue regeneration, we can see that we need to have chemical signals, I guess, that are sent to the area to activate this regeneration. So if all goes well, if the injury, let's say, has not been too detrimental, then usually we will get regeneration. We will get the damaged cells and damaged tissue replaced, but sometimes that doesn't always end up that way. We could end up with some scar tissue in most instances, depending whereabouts on the body that has occurred. It's not, life-threatening, but unfortunately with certain organs, which we're going to move into in our next topic, it can be life-threatening when certain organs are scarred. This regeneration process is quite self-explanatory. So hopefully we'll get this new growth, and that new growth of cells and tissue should completely restore any part of the tissue that has been damaged to its normal state. So that's really, really important when we look at tissue regeneration that has been successful, is that we're looking at that tissue being restored to its normal state. So we go back now to our stem cells that we learnt about way at the beginning of the semester, and they play a big part, the stem cells, in helping make new cells that will then regenerate the tissue. Having a look now at replacement. So what is going on here? So there's different phases that the tissue actually needs to go through, that depending on how much that tissue has been damaged, it takes for the full recovery, I guess, or the full replacement to be done. So we're looking at healing of tissue. So it needs to completely restore some part, depending on the organ or tissue that's damaged, of that tissue, or tissue that is non-regenerable. Okay, so we'll have a look at non-regenerable soon. So these are the phases, and try and think of an example. I'm sure everyone's had an example where they've had an injury or an operation or something like that, and you may have seen some of these yourself. So try and think of that example on yourself as you are going through this. So usually in the first few hours, that's when we'll see, you've fallen over and you've scratched yourself or whatever. We see the bleeding stage. Okay, so we get that blood that obviously is near the surface of the skin anyway. This can also be happening internally, by the way. So if we're looking at a kidney or pancreas, something like that, then this same bleeding, so it leaks out of the blood, will leak out of the tissue that's been damaged, whether that be internal or external, so on our skin, for example. So we'll get that bleeding. If you've done your first aid, which I'm assuming you did in year 10, you would have gone through how you would stop bleeding in a situation where someone has injured themselves. So you need to put pressure on there. And what we're trying to do is get that first stage I mentioned a few minutes ago on the blood sort of clotting there. We want that. We then start to get inflammation. So if anyone's had quite a deep wound at any time, or a good example is if anyone has had an operation on anything before, you'd know you get quite swollen around that area. So it's quite inflamed. And that is, again, going back to that first slide we did, is all the macrophages, neutrophils, all those white blood cells that are rushing to that area to start to repair, I guess, and make sure with that bleeding, obviously a surface has been broken. No pathogens have hopefully gotten in, so they've got to clean that all up. And then we get the proliferation. So we're now moving into weeks, depending on the type of injury it is. So usually we'll get the bleeding and the inflammation. If you look at that little image there, they usually occur within minutes, hours, or days. Okay. Then if it's quite a deep wound, this is going to change depending on the injury. We get the proliferation, which could take weeks. It could take months if someone's had an extremely bad, accident in sport or something like that. They need time to heal. We get this proliferation of stem cells making the new cells that we need for the tissue, and then we get into the remodeling phase. If anyone's had an injury, it could be you've sprained or torn a ligament or a tendon, or it could be a broken bone, or it could be anything like that. You know that it could take weeks or months just to get yourself back into the same active state that you were in before the injury happened. So again, small cut, that tissue should repair pretty quickly. quite a major, accident or surgery or break then in a bone, then that could take months. Process in a bit more detail later, but just quickly, the main process here is we get this new connective tissue that is laying down, and that's known as scarring. Sometimes we can see our scars, so we might have, if we've cut ourselves or something like that, and if it's deep enough, we might get some scar tissue that's been left there, or if you've had a surgery and they've had to stitch you up afterwards, there could be a scar that has been left there as well. So if the tissue is able to repair really well, then the scar tissue can eventually sort of, would they say fade, but I don't know if that's the right word. It generally, it gets replaced, let's say, with new, more functional cells and tissue. But if it's quite a big scar that's been left behind, could be in your muscle or a ligament. I remember doing my ankle years and years and years ago when I was still competing in sport, and I tore all three of the ligaments off my ankle, like they actually came off the bone. And there was a bunch of scar tissue that ended up developing, I guess, around those tendons. So I had to go through a bunch of physio to try and break down the scar tissue so your ankle can get back to normal function. So it's quite a common thing. If we have tissue damage, unless it's minor, like if it's minor, then it just regenerates and it's done. Otherwise, it consists of both regeneration and replacement. And usually with our epithelial layers, and that's not just our skin, it's the layers that are around our organs and things like that, can be restored to most of its original layers. It just depends. We've spoken about scarring or scar tissue that might not be life-threatening, for example, but there are some organs that it can be. It can be very detrimental to your health. It could actually cause, death. Having a look at our cardiac muscle. So remember our 3 different types of muscle, and they all regenerate and restore themselves differently. So our heart or our cardiac muscle, this is really, really serious if we end up with a scar, any type of scarring in our cardiac tissue. Our cardiac tissue, as you can imagine, because it's constantly beating, and if you remember back to the dissections that we did, and we had the heart, and if you compared that to the other tissue, it was quite strong. It was quite strong and hardish, I guess. It needs to be continuously beating, 24/7. So if that then something has happened that has damaged the heart and left a scar, then it's going to absolutely affect its function. It's not going to be able to contract and relax to pump that blood around as efficiently as before, and that could lead to things like heart attacks and all sorts of things. Myocardial infarction, that's what that is. So structural abnormalities could lead to that organ just not working anymore. So you end up with this dead heart muscle. So having a look at the image there, if you look down the bottom, or look at the heart, picture of the actual heart, down the bottom it's like this brownish colour. You can see that is very different to the rest of the heart, and you can see that that tissue is not going to be working. It's dead. It's dead heart muscle. It can't pump that blood up. It's already trying to work against gravity there. So it's blocked. Looking at the second image, so B, you can see the healthy tissue and then the dead tissue and how it's blocked there by something. We got plaque buildup that's there from probably an unhealthy diet and smoking. Let's just add vaping in there, why not? So those sorts of things that end up blocking the tissue or the artery then ends up killing off the tissue. And if your heart stops, that's it, guys. as we'll see, are more capable of others to regenerate via cellular proliferation. So getting more of those stem cells. So we'll have a look at that next. Different types of tissue as far as regeneration goes. It's really important so you know which ones are most likely to regenerate and which ones are not. So we have three different types. We have ones that are continuously dividing all day. So they're being lost and replaced quite quickly, skin cells, etc., blood cells, those types of things. Then we've got our stable tissue or quiescent tissue, never say that word. So that has a minimal proliferative activity. It can proliferate, it can replace, but it's not as prevalent as the continuously dividing tissue. So then you've got your smooth muscle, things like that, endothelial cells, fibroblasts, and usually that only happens when there's been an injury of some sort. And then you've got your permanent tissue, so it doesn't proliferate after birth. So terminally differentiated, it's done, it's gone. And that's when you'll end up with scar tissue. So neurons, we get that throughout our nervous system sometimes, our cardiac muscle we just looked at, and also our skeletal muscle is very easy if you have an injury of some sort and don't go to a physio and do as they recommend, you could end up with scar tissue there too. Continuously dividing tissue. So I said before that they're constantly dividing and they replace dead cells. So our skin, etc., salivary glands there, your gastrointestinal tract, that's always being damaged and replaced. Inside our stomachs, inside our intestines are always continuously dividing. So mitosis is going on there all the time. Cells, for example, with a skin cell here, so they've got a really big ability or a greater ability to proliferate, and they can self-renew and self-regulate pretty quickly, so we don't really need to think about it. So if we're losing a blood cell or a saliva cell or whatever, then it's just going to be replaced and that's that, and we're done and off we go. But it can also give the continuously dividing ones can also give rise to more than one type of cell. We're looking at the shape, so the asymmetrically, the shape of it as well, not just the structure. And then it gives rise to one daughter cell. So we got the mitosis, basically, it's differentiating and maturing. The undifferentiated cell, so the original stem cell that is still going to be there, that remains in that state, so it is capable of self-renewing other new cells. Type of cells, the second type of tissue regeneration we have here, the stable tissue. So these are ones that can reproduce and regenerate when they need to, as if they're injured, etc., but they're not doing it all the time like our skin cells. So using my favorite of the organs, the liver, there's a lot of stuff going on through our liver all day. The other great thing about the liver is that it does actually regenerate quite well. If we're generally looking after it, our liver, that is, and what's going into our body and toxic substances and those sorts of things, the liver's pretty good at regenerating itself. So it's getting a lot of cytokines or growth factors that will go into the liver to regenerate. You get a lot of differentiation, a lot of regulations. It's quite regulated. You can imagine with all the toxic filtering it needs to do all day. When we have, and at the very bottom of the liver image, you'll see that there is persistent damage at the bottom there. So if you're constantly, an example of that is drinking too much alcohol or taking too many, drugs or whatever. So you're constantly putting your liver under stress and you're constantly basically asking your liver to regenerate. It's got other things it needs to do. It doesn't have time to just, at the drop of a hat, keep making new cells willy-nilly. So it needs to do better things than do that. So usually your liver is quite healthy if you take care of it and you're not making it do extra things. So next to persistent damage, if you look to the left of that, we have prevention. So looking at not, doing that. It's not about not ever having a drink of alcohol or something like that. It's about not doing it consistently all the time. That's when we're going to have this persistent damage, which will be a lot harder for the liver to then make more tissue. That's also occurs in parenchymal cells, so things like, I said the liver already, but our kidneys do this as well, and our pancreas. So with those two, you can imagine as well, kidneys are very important when we look at detoxifying and filtering through our blood. So if we're putting our kidneys under extra pressure to do extra filtering that it really doesn't have time to do because it's got other things to do, then it could end up with damage to the kidneys and pancreas as well, because they're basically containing our islet cells and making, insulin, etc., etc. So we don't want to end up overloading our organs with other things that we are putting in to ourselves that our body over time won't be able to handle. Dividing tissue. So I've kind of mentioned this before. I have no idea why I put that meme there because it doesn't even go with what I'm talking about, but I guess that's like something I would do, I guess. So it's known as permanent tissue. So things like our cardiac tissue. So they've already left the cell cycle. They've already left the sort of stem cell phase. They're not proliferating constantly. And it can lead, usually always, will leave a permanent injury like a scar or something like that. Our skeletal muscle, we'll look at that. So at the very top of that image, we can see a normal muscle structure. The muscle fiber, we have our satellite cells that are really important when we're looking at building more skeletal muscle cells. We've got the motor nerves taking messages from our brain down to our muscles, and that should be working fine. If we go to the right, we look at the arrow going down there. We've had some kind of damage to the muscle fiber, normal wear and tear it could be, or it could be, you've gotten an accident and cut yourself or something like that. Then it's caused a bunch of damage there. Coming down to the bottom of the picture, we've got our satellite cells. So again, they're very, very important in our muscle cells when we're looking at regenerating. Anyone who's trying to build more muscle, either purposely or they've just got an injury and need to repair it, the satellite cells are the ones that do this work here. So they proliferate and they realign, start building new skeletal tissue. And then hopefully they are matured and regenerated and off they go. But it doesn't always work that smoothly. There could be some reason why a scar has formed within that tissue, but it already looks different to the top picture. Here with the tissue regeneration, we've got continuously dividing. They are really, really good. They're excellent at renewing themselves. They undergo lots and lots of cell cycles over a lifespan. It doesn't happen in the parenchymal cells, so they are the ones that have already differentiated. It happens in stem cells, which are deep inside the tissue. Somatic cells, they are located, so they're in microenvironments. They're in little microenvironments within the tissue. They contain both stem cells and non-stem cells. And the neighboring non-stem cells signal, they send out a chemical signal that tell the stem cells they need to divide when they are needed. Learning by osmosis. Wouldn't that be good if you could just lie on top of the encyclopedia and just absorb, let that knowledge diffuse through into your brain? That would be much easier, but alas, we have to actually learn it. Okay, so what have we got here? So we've looked at now why and the types of different regeneration. So we're going to look at the repair of those tissues now. So we've got 3 critical types of cells for the tissue repair we're talking about now, not just regeneration, repair. So we need some fibroblasts. Let's chuck some of them in there. What about some endothelial cells? Yep, need those. And also our macrophages. We also need a bunch of macrophages. First things first, we've got these fibroblasts. They become activated and their job is to lay down fibrous tissue. So that then moves to the area that's been injured. So you can see this little picture over here that the fibroblasts are activated. So we've got some physical stimuli that start to produce the collagen fibers, and then you've got your chemical stimuli. So that's when it's sent to a receptor to start the repair phase. After you get the fibroblast, you can see in the image right on your right, of the PowerPoint, that we have the fibroblast. They're those little purple dots that it's pointing to there, whatever color that is. So they're laid down first. We also have some elastin. So if we're looking at skin tissue or epithelial tissue for our skin, we need it to have some elastin in there as well, so it can move. Or think of actually fingernails, same thing, hair, same thing. And then we also need some collagen fibers in there as well. So that helps to provide the structural component. And then that ends up building the fibroblast to keep it nice and strong as well, forms those new tissue cells, and then our elastin fibers. This is a good image actually, so we can see sort of what type of injury needs the most amount of remodeling or tissue remodeling there. So to do that, once the injury occurs, we have these growth factors and cytokines. We'll look at both those later. So the fibroblasts are then suspended together within these new blood vessels. And then we start getting granulation tissue, which is the first sort of laying down of what hopefully will be the new tissue. So growth factors and cytokines suspend those fibroblasts and we get granulation tissue. So that's happening relatively quickly. So you can see then with the inflammation stage, it's straight away, like it's as soon as the injury happens, we have that going on. And then we get our granulation tissue over the upcoming days, depending on the type of injury. And then we move along, could take a month or more. We start getting the wound. It should be able to contract at this point, and that's why it's important. So we don't end up with scar tissue. So then we get the collagen that accumulates and the elastin fibers that I was talking about, which will allow the tissue to go back to hopefully close to normal as possible. And if that doesn't happen and we don't get our wounds being able to contract afterwards, that's when we get the scar tissue that builds. What's going on here? Right, so I've given a little bit of background here. So new blood vessels need to carry nutrients, and new blood vessels need to form, whether they're capillaries or bigger than that, to get cells to the new tissue. They then get those fibroblasts, but the rest of the process doesn't happen. So it leaves a scar, and at this point when it first happens, the scar is collagen. It's just collagen, and hopefully the scar tissue can be remodeled and strengthened over time. So in most cases that happens, but sometimes it doesn't. We're left with the scar at the end. Earlier, the growth factors that have to be secreted. So where do they come from? Okay, so we get these macrophages. So we're going back to the immune system here, and it says antigen presentation there too, which we normally think of a disease, but think of anything that can injure the body in some way. That's what we're looking at here. So we're looking at stimulating, the growth factors then stimulate the fibroblasts. And then we get these, so keratin in skin cells, for example, they start depositing new cells and remodel throughout the extracellular matrix there. So we've got the growth factors. You can see it all down in those images. I'll let you have a look at those yourselves. Looking at the scar tissue formation and our wound healing. So next we have this the migration of those cells. So they may not necessarily all be made, I guess, or replicated at the site of injury. They may be replicated somewhere else and travel somewhere close by and then travel through our blood. So once that happens, it lays down this extracellular matrix and we get the remodeling of the collagen to form a durable scar. So you can see in the image, you don't need to know this detail, of course, if you're going on to study medicine or something like that after school, then yes, you will need to know how that's happening on a chemical level, but not at year 11 level. So we're just looking at this is what happens, this is where it starts, and that's what's happening when it finishes. We get collagen and a durable scar. So we don't want to walk around with our wounds just reopening all the time. I mean, that's pretty gross. So we want it to be a scar that's going to stay closed. Things that are growth factors and our cytokines. They're called ligands, but you again don't need to know all the biochemical components of this. So just know them as growth factors. And these cytokines float between our cells. And what they do is carry information around the body. So they're carrying information maybe to say, right, we need some new cells here to form this tissue because of this injury, etc., etc. In this instance, once the cells receive those instructions, the ligands or the growth factors bind to our cell. So they bind to the surface of our cell and they have little receptors there basically that allow the signals to flow through with the instructions. Big one. So what happens then? We get some actions that are initiated. So the message is then brought into the cell, and then that message results in a new action. So it could be the migration of new cells, the proliferation making of new cells, some kind of secretion of a substance that's going to help aiding the development of the new tissue. They're peptides. So poly means many, so many peptides, usually a chain of those molecules that bind to the receptors on the cell, delivers the message. And having a look at the image on the side there, you can see that it takes the message. If you look at the top part of that image, we've got a growth factor that comes along. So that's that little circle thing there. And it attaches to the receptor, which is that orange looking thing on the surface of the cell. And that then takes a signal, which is in this instance, tyrosine kinase start signal basically, for the cell to start dividing. And when it divides into two, then it's forming a new cell to replace the cells that have been injured or depleted, I guess, in the tissue before in the injury. Growth. There's a lot of biochemistry that goes on behind this, but we're just going to do the shortened version, let's just say that. So we've got our transforming growth factor, the beta, made by platelets. So you're looking at our blood in this instance, our endothelial cells, our lymphocytes, and our macrophages. Again, blood cells, those last ones are white blood cells. They also bind to the surface of our cells. So they have little receptors. All of our cells have, for whatever substance it needs to function, has its own little receptors to allow certain messages in at certain points in time. And once the message is in there, again, it triggers this transcription factor. You don't need to know at this point these SMADs, SMADS, that just know that is a process. You will not be tested on that, but that enters the nucleus and then that starts that gene transcription. It starts the making of the new cells that we need to form the new tissue. That fibrosis, I've got an example here. We're looking at a mouse there and looking at the fibrosis that can occur in those images. You can see the control. Then you've got 2 months, 6 months, and then 12 months. You can see those little kind of white circly things that are in there, or beige coloured ones. They're fibroids, so fibrosis. So it attracts fibroblasts to start proliferating. Then they secrete collagen. What that collagen does is inhibit the degradation of the extracellular matrix. So this is then moving on to, if it doesn't work properly, how we end up with scarring. Because if this isn't continued, this process isn't continued and continued along the lines it should be, then we get this excess collagen that is secreted. The extracellular matrix will stay, and then it can then form hardened tissue. But if we're looking at wound healing in general, this is our first phase, and then hopefully after that, the rest of it goes as it should. The first slide, of the one previous to this one, looking at fibrosis again, and we've got my favorite, the liver, back again. So it inhibits epithelial cell growth and it limits inflammation. Inflammation at the start of an injury or illness is actually really, really important. Okay, we need that because that's all our blood cells, our white blood cells, our immune cells that are going to that area to fight off anything that shouldn't be there. So it's only a bad thing, inflammation, if it continues and it stays like that. But we want inflammation, so having it limited is not a good thing. And then what it does is promote invasion. It can also lead to tumours as well. So having a look at the things that can lead to that, we're looking at fibrosis, having a look at the image, it could be from the unhealthy diet, alcohol, chronic infections, for example, that then if it's not changed, can lead to chronic liver injury, and then we get to the liver fibrosis. So they are those hardened fibroids that end up in our tissue that shouldn't really be there. They should repair basically, and then tissue goes back to normal. But by constantly having either these examples going into your liver, I mentioned this earlier in the presentation, it doesn't give your liver a chance to really replenish properly, regenerate properly. And we don't want it to go past that fibrosis stage. Okay, if we can get it to reverse even before then, that's a good thing. But if we start getting to liver cirrhosis, so we start getting death tissue, so liver tissue death is liver cirrhosis, usually that will end up in unfortunate circumstances, let's say. Again, to go into this in detail, there's a lot of biochemistry, so we won't. At this point in your education, we'll leave it at the basics. So these are released on platelet activation. So once the platelets are activated, so that gets those, it sends out a message to instruct the neutrophils and the macrophages and the fibroblasts to move to the wounded area. Okay, we don't have them just sitting around all the time, otherwise our body wouldn't function properly. It waits for a signal and it waits for instructions to move to where they need to go when they need to go there. So they stimulate, activate these, and then they get that production of a matrix. So we're looking at getting that structure or architecture into our new tissue. Matrix is also really, really important. It's a special type of tissue, or it helps to form special types of tissue. And it's important that we have this extracellular matrix when we're looking at tissue or wound repair, because it gives resilience to our soft tissue. It also gives firmness to our bones. So if you think of a matrix of, how is something structured, it's like it's got its own scaffold there to keep the tissue, bone, whatever it is, that it's there to repair in the right structure so it can function. By doing that, it leaves it open enough to get new cell growth in the tissue. We got our proteins that come along, good old proteins, the collagen. That's important to give strength to our tissue. And I've mentioned already the elastic fibers, so we can stretch and snap our tissue back into place. And when I say that, I don't mean literally snap it. I mean that it can stretch. If you're doing sport or some kind of movement, you want your ligaments, tendons, whatever it is, skin, I don't know, to stretch, but also to go back into place once it's finished. Fibronectin is very important again when we're looking at wound healing. So this stabilizes the blood clot. That's why it's really bad when people go around and just pick off all their scabs or whatever, and then it starts bleeding again, and then you got to go through the whole process again, and all it's doing is prolonging the wound from being repaired. So the fibronectin that comes along to the initial blood clot that fills in the wound, and it binds to fibrin that's there in our skin to stop the bleeding. But it's also important because it gives a framework for the collagen matrix to be built. And that has to happen for wounds to heal properly. So again, if you want to prolong your wound healing, then pick away at your scabs. But if you don't want to do that and you don't want an infection, then it's really important that you leave it as it is. Let the fibronectin do its thing, let the collagen matrix do its thing, and then get our wounds healed and back to normal, hopefully. We have regenerated our tissue, or it has scarred. So if we know that it has regenerated, we can determine, or a doctor, a surgeon can determine that the tissue has regenerated when the new cells replace the damaged or dead cells to their original state and does not occur regularly in humans without scarring. Okay, so the tissue architecture and function after an injury. So we've got our regeneration of our injured tissue. Then we get replacement of connective tissue, scarring. This form of scarring is what we kind of want, right? Then the tissue repair usually tissue repair involves both processes. If all is well, the scarring will then have new functioning tissue cells and tissues replace it. If not, we get a permanent scar. Cells need to be capable of proliferation for this to happen, and there has to be underlying stromal framework that's intact for this to happen. Here's a nice image, if you want to call it that, of some tissue damage and scarring that's happened in someone who has had an injury. So you can see where the injury is going there, right on the sort of tendon ligament area that's attached to the bone and the muscle. So if there's severe tissue damage, the framework underlying, then and it happens in non-dividing tissue, that's where we get the scarring. So have a look at the close-up image, I guess. You can see the difference and to the structure. Basically, they're not nice, smooth, skeletal, striations there. They've got these white lines, irregular lines flicking through it. So that's causing scars. And whatever has happened, whether it's regenerated or it's been repaired, that depends on the cells. So how well they can proliferate, how well they can do it, the capacity of that, how good the stromal framework is, how good our inflammatory response is, and how long the injury lasts. It's all going to have an impact on how or when our tissue regenerates and or repairs. You can have a look at that image there. So that one's for sort of microbes. We don't want those getting into the wound, which again, I go back to my disgusting scabs I was talking about before. If you pick them off, then those things are going to get in, and that is not good. With our overall repair, we get this influx of debris removing inflammatory cells. So getting rid of, anything that shouldn't be there, any pathogens that have gotten in due to the injury, basically, and they get it to the connective tissue. That then forms granulation, so tissues of fibroblasts that converts it to granulation tissue, which goes then to fibrous tissue. And then, if all goes well, it's remodeled over time to form a scar. And again, have a look at the image there yourself. Pause the recording if you want to have a look at that process in more detail. To summarize real quick, the 5 main components of tissue repair. We've got here inflammation, new tissue formation, fibroblast proliferation, collagen synthesis, and scar remodeling. And you can have a look at this image. You can see in the first one at the very, very top, the first image, we've got a wound that has occurred. It has caused damage to our skin coming along there, depressed scar. So lost tissue or collagen, you can see there. So there's like a little dip in the skin there. Having a look at the third one, a raised scar starts to form. So we've got, it forms basically overgrowth of collagen. Then we come down to the bottom here, so accumulation of keratin and sebum, so a little bit of oil with the sebum there, and the keratin gives it nice strength. Looking at bacteria proliferate, so get rid of those by forming the scab, and then we get that marked inflammation, and then it goes on from there.